Welcome to a very special episode of Corey's Chromosome Chronicles. This is your frequently asked questions section. This is where you guys have sent in some questions about a topic of interest to you, and I'm going to answer them the best I can. This week we're looking at cells, the cells topic in SACE Year 12 Biology, and we've got a bunch of questions that have come in. The first one has to do with the relationship between photosynthesis and respiration. This is a really important topic in Year 12 Biology. It's something that is quite difficult to answer, but something that you should be able to grasp anyway. The first thing is make sure you know the equations. Know the word and chemical equations. The word and chemical equations you'll see above my head at the moment. For respiration, remember you're talking about the breakdown of glucose. So you're talking about C6H12O6 plus 6O2 going to 6H2O plus 6CO2 plus energy. That energy bit is really important. Don't write ATP. I know it's easy to do because you think that it's a chemical equation, but remember ATP does not come out of respiration. ATP synthesis is a separate process altogether. Photosynthesis is the exact opposite, so that's what I mean by relationship. Photosynthesis is a synthesis reaction that allows for the making of glucose, which can then be broken down through respiration. So the equation for that is 6H2O plus 6CO2, along with, remember that on that arrow, you need to put in sunlight and chlorophyll, goes to C6H12O6, which is glucose, plus 6O2. So it's the exact opposite equation, and that's how they relate to one another. Remember, photosynthesis is a synthesis reaction, but it gets its energy from sunlight. It then creates glucose, which can then be broken down in the presence of oxygen, if we're talking about aerobic respiration, into carbon dioxide and water to release a fair bit of energy to allow you to do ATP synthesis. What sort of questions could you get on it? You could get a whole bunch of questions relating to the equations and the process themselves in terms of where they occur. If you're going to look at the relationship between them though, which I think is what the question is about, then really you're going to get a graph which you can see up on your screen now. That graph shows oxygen input and oxygen output on the y-axis, and then there's light availability on the x-axis. Light availability is generally what you'll find on the x-axis because that is going to affect your rate of photosynthesis, but it's not going to affect your rate of respiration. Now in terms of a plant, respiration is always constant, so it doesn't matter whether it's day or night, it's going to happen around about the same rate. But of course, because photosynthesis requires sunlight, it's going to increase the rate of photosynthesis during the day, and then the rate of photosynthesis is going to decrease as you get towards the night. Now that's going to affect the oxygen input and output that you would detect from a leaf because of the fact that respiration and photosynthesis require oxygen on different sides of their equation. So let's have a look at point A here. We've got an oxygen input, quite a large oxygen input, but you can also see that we have no light there because we're right on the left-hand side of our x-axis, and that makes sense. Because if there's no sunlight, it means that there would be no photosynthesis happening. And if you remind yourself of your equation, respiration requires oxygen. Because the leaf wouldn't be making any oxygen through photosynthesis, it's having to take an input of oxygen from the outside environment. If we have a look at B there, you can see that we have no oxygen input or output. What that means is that the rate of respiration must be equal to the rate of photosynthesis. In other words, there's enough sunlight to allow photosynthesis to happen at a fast enough rate, that it's producing enough oxygen that it can supplement the oxygen required for respiration. So the leaf is not either taking in or releasing any oxygen. Now, if we have a look at point C, now point C is where a plant would be generally during the daytime. It means that the leaf itself is producing quite a lot of oxygen itself and releasing it from the leaf. It's doing so because the rate of photosynthesis, or the rate of oxygen production, is much greater than that required for respiration, so therefore you're getting a net output of oxygen. Now, is that the only question you're going to get in terms of relationships between respiration and photosynthesis? No, it's not. There are others. They're generally graphs, but they all have to do with amount of glucose and oxygen and carbon dioxide relative to each other, generally talking about in the daytime or the nighttime. So if you see a question like that, know that it's referring to the relationship between respiration and photosynthesis, and then you should be able to work it out from there. The second question I received has to do with osmosis, specifically the idea of the solute concentration. Now let's start looking at osmosis. Osmosis itself is just the diffusion of water. It's the movement of water from a high concentration of itself to a low concentration of itself, passive. So it should be really easy. So why do students find it hard? Because students find it hard to talk about the solute concentration, which is what we generally do with osmosis. The idea is, if we're saying the solution has a high water concentration, inversely it must have a low solute concentration. Because in a solution there are only two things: the water, which is the solvent, and the solute itself. So saying high to low water is the same as saying low to high solute. I think the best way to explain this is to look at a question. So let's look at the question that appears above my head, which I'm going to read off here. Fish have an internal salt concentration of 3%. If placed in the ocean, which has a solute concentration of 5%, the cells of fish would A. Gain mass and increase in surface area to volume ratio, B. Lose mass and increase in surface area to volume ratio, C. Lose mass and decrease in surface area to volume ratio, and D. Gain mass and decrease in surface area to volume ratio. Now, there's two parts to this question, and that's why it's a good question I want to talk about here as an example. Surface area to volume ratio heavily relates to osmosis, because if a cell takes in water, it's generally going to get larger, and remember with the surface area to volume ratio, larger objects have smaller surface area to volume ratio, and that means they're less efficient at diffusion. Whereas, if water is drawn out of a cell, it's going to generally decrease in size, which means it's going to increase in its surface area to volume ratio, and it becomes more efficient at diffusion. So these two theories generally appear together in questions. Let's focus on the mass bit first. The best way to answer a question like this, instead of trying to just think low to high solute and high to low water, is to just draw yourself a simple beaker. So if you draw yourself a beaker with some water in it, draw a big circle in the middle. That means your cell, or your fish, or your tissue, or whatever it is that the question refers to. Now in this question it says that the fish has 3% internal solute concentration, so write a 3 inside of your circle. The external concentration, or the concentration in the ocean in this case, which is in this case the liquid in your beaker, is 5% solute concentration. So write 5%. Now, you know the water is going to move from low to high solute concentration. So it's really easy now to see on your beaker that the water is going to be drawn out of the fish because it's being drawn out of your circle and therefore out of your cell. So in other words, the fish is going to lose mass. Now again, in answering this question, let's have a look. A was gain mass. That can't be right. B is lose mass. That could be right. C is lose mass. That could be right. D is gain mass. That can't be right. Now we need to think about surface area to volume ratio. Remember what I said before. If an object loses mass, generally it gets smaller, and therefore its surface area to volume ratio is going to increase, because smaller things have a larger surface area to volume ratio. So that's what we're looking for. So if we go back to our points here, point B is going to lose mass and increase surface area to volume ratio. We now know that's the right answer, so the answer is B. I hope that helped with osmosis. Our next question refers to the fluid mosaic model. Now the fluid mosaic model is a model which explains how the cell membrane works, and it should be fairly easy. There are two parts to it, the fluid bit and the mosaic bit, and you are allowed to break it down into those two sections in your answer. The fluid bit relates to the fact that the cell membrane acts more like a fluid, where the molecules can constantly change their position amongst one another. The mosaic bit relates to the fact that the cell membrane is a mosaic of different types of proteins and other molecules. The mosaic word means it's made up of many different parts. And it's as simple as that, and you can answer it in that way. Our next question has to do with different types of proteins that embed themselves within the cell membrane. Now there are 4 types of proteins that Safe Stage 2 Biology want you to know. The first one are carrier proteins. Carrier proteins are proteins which can carry molecules against the gradient. So carrier proteins are shut off at one end, they use energy, or the cell expends energy, to draw molecules in and actively pump them across the gradient from the side that has lower solutes to the side that has higher solutes. So this requires energy, and it's related to active transport. Channel proteins are pretty much like their sound. They're proteins that just act like a channel, like a doorway within a classroom. They allow molecules to passively pass from areas of high concentration down the concentration gradient to areas of low concentration. Okay? They're there to allow molecules that can't move through the membrane freely a bit of a doorway to move through. It's as simple as that. Anchor proteins are proteins that, like they sound, anchor the cell membrane itself to the cytoskeleton and the cell membrane to the extracellular matrix. So in other words, they anchor the cell in place. The last one is receptor proteins, and these proteins are ones which receive signals from external stimuluses, so they might receive signals from hormones, say a growth hormone. They have a site, a binding site, which is specifically shaped for a type of hormone. When that hormone binds to that site, that then triggers the protein to send a message into the cell, transmitting that message. And it's as simple as that. They're the 4 different types of proteins you need to know. Channel proteins would look like a channel themselves. Carrier proteins would be shut off at one end. Anchor proteins would look like a little anchor. And receptor proteins would have a specific binding site, and they probably wouldn't be embedded all the way through the membrane. So I hope that helped.